Well, good morning everyone and welcome to sunny Forthside. It's so great to see so many of you here this morning, and it's a real privilege to be able to showcase what we're doing here at the farm. And it is a wonderful annual event, and there's so many things we'd like to take you through over the next few hours, and you're going to hear some great speakers and some really great farm walks as well, and you're going to see the farm in action. And it's a real privilege to be here to do that. We're here now to really spend some time thinking about the farm and foresight and horticulture, and really celebrate what it's all about. The event is designed to be a day of learning, a day of catching up, industry engagement and celebrating our farm. And we're going to learn about our latest research, our insights that shape Tasmanian horticulture. Our presenters are certainly going to welcome questions, welcome discussion, welcome conversation throughout the morning and also through the field walks as well. So please don't hesitate to ask. Please don't hesitate to challenge them and have conversation about it. So today's event brings us updates on our net zero emissions farm. We're going to hear from Tory. You'll hear about the latest in mop-top research, the mop-top activity that from Calum and Doug. You're going to hear from the latest on our irrigation project from Dan and the economics of that. And you're going to hear about our pyrethrum research from Tamieka. And from that, we'll have an engaging panel discussion on biosecurity and some of the challenges around that. And we'll hear from someone from Biosecurity Tas, we'll hear from our new biosecurity staff member here at TIA, Craig Elliott, and also Doug and Calum as well. And then we'll go for a walk across the property where researchers will be able to show their insights and address some of your questions. But without further ado, I'd like to invite the Minister to the stage, Gavin Pearce, and to say a few words. Thanks, Gavin.
Well, thanks, Nigel, and good to see so many smiling faces in the room. It's a really refreshing sight for me coming from Hobart this morning, but I feel as if I'm home now, and it's great to be amongst friends. This is, of course, my home, the Northwest Coast. We've been farming here for 6-7 generations since the mid-1800s. I'm very proud of it. I think it's the best place, not only in the state, but in the country. And we've got the very best farmers and agricultural scientists behind that vision. So I mean what I say. I feel as if I'm home and I'm happy about it. Just reflecting on what Nigel said, I think it's an exciting day. I think it's, we're all gathered here. We're all, we share that vision. We share that excitement, but today is how we put that into place. Today is how we put that science through that dashboard into the hands of farmers who make that happen. And I think that mechanism is the important thing. That's the thing that I concentrate. I understand that we've got some of the best researchers in agriculture anywhere in the world. I really get that. It's been borne out to me, and some of the dealings that I've had to do already in my job as Minister. You remember day one, it was mop-top virus. Now, I was surrounded by the absolute subject matter experts, not only in the country, but in the world, and they're right here. And they reassured me, they reassured that farming community, they reassured that industry, they reassured the rest of the country that we're on top of this quicker than anybody else could. And it's an absolute credit to not only BT, but it's a credit to TIA, all your researchers, and it's a credit to the industry. Remember what I said about that relationship that industry needs to have with science? That dashboard, that's how I refer to it. That is absolutely crucial. And I see that every day in the work that TIA is doing. And I know Mike shares that vision. And I know Mike understands just exactly, and his researchers get that they can't hide their light under a bushel, that they need to share that with the grassroots farmers on the ground, and that's important. We've got a good story to tell. We really have. In fact, in the last report from ABS, agriculture, forestry and fishing grew in Tasmania by a massive 6.7%. That's the 24-25 numbers. It's probably due to the aquaculture numbers, and I happen to have, you've heard that I've got salmon to look after as well in the southern part of the state. It takes up quite a bit of my time. But that's a $2 billion plus industry. It's worth as much as our dairy and our beef put together. That is agriculture, it's aquaculture, but it's certainly a great contributor. The other sector is dairy. and dairy continues to perform. It continues to grow. It continues to thrive, and it also continues to be an early adopter of R&D. And TIA play a very important part of that. You know, that Elliott research station and the job that we've done there, it's an absolute credit to you, Mike, and your team. And I look forward to visiting Elliott very shortly. In terms of TIA and the support that we provide from state government, now this year TIA will receive more than $5.1 million. $5.1 million that I think is well spent, that I think is probably going to be multiplied not only in terms of a dollar value, but in terms of long-term strategic benefit back to industry. It's all very well and good to throw money at something that stays contained. It's when that money is delivered, is inculcated, it's sent down to the lowest possible levels. When I say lowest possible levels, I mean back to that person that's on one of those big green things out in the paddock. That's what's important. That's how I measure it. That's how the industry measure it, and that's how productivity happens. I think if we thought more about that, then the world would be a greater place. And I'm glad to see TIA holds that vision the same way that I do. In terms of the Agriculture Innovation Fund, we've put 1.6 million over 3 years in there. You know, we've provided 250,000 towards rapid detection of pathogens in pyrethrum. That's important. State government has also backed net zero emissions horticulture, and it's demonstrated right here, and you'll see that today, with another $462,000 in funding. We've had to put 200,000 into mop-top and also the powdery scab. And a big shout out to Doug. Where are you? Doug here? He's hiding. He's probably digging spuds. But the work that he's doing, the work that that team is doing, the work that TIA is doing in relation to Spongisporia subterranea, powdery scab, and the control of that particular vector in managing mop-top is incredibly important. It's vital. It's not only vital for Tasmanian farmers, it's vital for the rest of the country. Because eventually they'll wake up that they probably have that virus too. So the work that we do here is absolutely groundbreaking. The work that you all do is absolutely groundbreaking. The reason that you're here today is to see that in action and to hear the strategic vision from Mike and his team on where the next step's going to be. Rest assured that I'm probably the most excited, most grateful bloke in Tasmania when it comes to be given this incredible responsibility that I have. I couldn't have picked a better portfolio. I couldn't have picked something that I love more. And I'm very proud that I'm in this position. I want to make the best of it. I don't know how many more years, how many more parliamentary terms I have in me, but I'll tell you what, I want to make the most of it. And I want to make the most out of Tasmanian agriculture. And I know you all share that vision. So that's why we're here all today. And I want to leave you with the fact that if we could get more people like you in this room, if we could spread that vision, spread that enthusiasm, go that dashboard that I talked about, and we can spread that right out into every paddock, into every farming operation, then that's what I want to see happen. I'm very pleased that we've got the catalyst for that happening today. So thanks for having me, and it's going to be a great day. I appreciate it. Thanks.
Thank you, Minister Pearce. Can I invite Mike Rose, our TIA Director, to come and give the Director's address this morning?
Welcome to the 2025 Forthside Open Day. It's really nice to see so many people here today, people from industry, here on what is a beautiful day, perhaps a little bit windy, but it's a really lovely day, and it's great to welcome you all here today. For those who don't know, TIA is a joint venture. TIA is physically located within the university, but we're a joint venture with the state government, with NRE Tasmania. And that means that we are strategically aligned with the state government and we work shoulder to shoulder to deliver for Tasmanian agriculture through that relationship. Put formally, TIA has got a mandate to deliver research, industry development, training and learning and teaching for agriculture and food within the state of Tasmania. Forthside is one of several facilities of various types that we've got around the state. Forthside here is a sandpit. It's a place where we can do research, where we can do testing, where we can experiment to see what works. And if it works well, then our job is to connect that with industry. And that is what today is all about, is to kind of give a roundup of what we've been up to on this site and otherwise, and then connect with you, our industry. Today is also a special day for us because we're launching our TIA strategy today. It's taken 18 months. As they say, it was a marathon, not a sprint. 18 months ago, we started with our staff at our staff conference and we took the last strategy and we just took it apart and we chucked out what we didn't like and we brought in some new ideas and we moved some of the furniture around. And then I had a first draft, which I took to industry and the TIA advisory board and to government and to all sorts of representatives from industry. And each iteration changed that draft substantially. So that what we finish up with today is genuinely a collaborative effort of how we can drive agriculture within the state of Tasmania forward. I want to thank absolutely everybody who contributed to that process. The strategy is a 5-year plan to take advantages of the advantages that we have and to address the challenges that we face here in Tasmania. We have many advantages for agriculture here in Tasmania. We've got our cool climate, we've got some of the most productive soils on the planet. We have really good policy support from government, and believe me, I'm genuine when I say that because I've lived in other parts of the world where that does not exist, and it is really nice to have that here. I'm just telling you straight. We have innovative, agile farmers that have shown time after time after time that they are able to adopt new crops and develop those and drive productivity forward. We also have a reputation for premium food as well. But we're not without challenges. We've got to get our commodities over the Bass Strait, and that is really expensive. We've got an incessant demand for labour, skilled labour, that we don't always manage to meet. We've got accelerating climate change. If you look at some of the predictions, and if we don't do something about emissions, then it really doesn't bear thinking about at the end of this century. Attendant to that climate change challenge, there are biosecurity risks as well. We've also got global market trends as well, and we've got President Trump with his tariffs, and actually that's to the benefit of the beef industry in Tasmania, and prices are really firm at the moment, thanks to President Trump. But predicting that is not always easy, and of course, where you've got something volatile, then it's very, very difficult. Our strategy addresses these things. We're going forward with optimism because I believe we have the right team. We've consulted widely, and by the right team, I mean the agriculture team in the whole of Tasmania, including TIA and everybody else. We have clarity of purpose. We have thought really carefully about how we best use that $5 million so that it is of best effect for Tasmania, and that allows us to walk forward with a great deal of resolve. Look, all organisations have a purpose, and within our strategy, our purpose is to deliver impactful research, industry development, education and training that supports resilient farming and food systems in partnership with industry, community and government. Every single word of that was debated and debated and debated within the leadership team and within TIA and otherwise as well. And every single word of that is meant. You know, Tasmanian agriculture is booming. To the 5 years to 2023, which is the most recent figures that I've got, annual compound growth is 8% per year, which compares to the growth of the Tasmanian economy more widely at about 1.2% per year. Tasmania is good at agriculture. Tasmanian agriculture is becoming more important to our economy, and indeed going forward, Tasmanian agriculture is going to be one of the key planks of our economic prosperity. However, research and development are still required. One of the advantages of Tasmanian agriculture is the state investment in irrigation, and I choose that as an example. But it's obvious that not all farmers are aware of what that irrigation, what that water can do for them, how it can transform their business, and that's just, I'm speaking anecdotally, but that's what I understand. And so therefore, going forward, some of the irrigation schemes that are planned haven't had the buy-in that perhaps they should do. There's a role for TIER here. The role of TIER here is to demonstrate the value of that water and then to connect that learning to the agricultural industry through our own efforts and then through private extension providers as well. As an example of a challenge that we might face, within 5 or 10 years, I'm told that Varroa will enter the state. And within 5 years after that, virtually all, as I understand it, virtually all of the wild populations of honeybees will be wiped out. Maybe not quite all of them, but virtually all of them. There are many agricultural businesses that depend on insect pollination, the apple industry and berries and cherries and so on, that don't move hives around, that depend on that wild ecosystem service, and they're going to have an alternative plan. TIER is working on that alternative plan. Equally, TIER is working on demonstrating the value of irrigation. You know, TIER adds value to the government investment. So the $5 million that the government invests in TIER, we add another between $10 and $15 million to make somewhere between $15 and $20 million total from other funders. And for every dollar of that research funding, so a research funding pot of about $15 to $20 million a year, over 10 years, each dollar of that is worth $8 to the agricultural industry. And that is a recent study by ABARES. Impact is critical to the work of TIER. There's no point in us doing research and it going on a dusty shelf and then that's the last that anybody sees of it. In fact, our mode of working is to start with the problems that industry face and then work backwards to the research that's needed rather than coming up with solutions and then trying to find a problem for them to solve. And so therefore impact, that change in industry practice to drive sustainable productivity is our goal, is our key performance metric. That success that we've seen requires continued investment in the JVA. And so it is with great pride and optimism that I launched the new TIER strategy. I'm told, yes, there we go. There's a QR code behind me there. And if you use the camera on your phone, and that will take you through to a website that will take you to the TIER strategy. And there you can read it in all its glory. Please use that strategy to work with us to deliver for Tasmanian agriculture. We are here to help you and to work with you. And we need that feedback, and we're listening. Work with us. Welcome to the Forthside Open Day. It's really great to see so many of you here today. And yeah, enjoy the day. Enjoy the discussions, enjoy the talks, enjoy the farm walks, enjoy the networking. It's really great to see you all here today. Thank you.
Thank you very much, Mike. For the next hour, we will now have our speaker sessions, and we will have 10 minutes for each speaker. Dan will come up and talk to us about our NRET-funded irrigation project and the economic component of that. Jon Finch will be talking to us about insects in agriculture. followed by Tamieka about her project on pyrethrum, and then Tory will be presenting to us the demonstration farm. And just before morning tea, we'll go out for a quick farm walk to have a look at that demonstration farm in action. Then we'll have morning tea and we'll come back here for some more speaker sessions. So without further ado, can I invite Dan to come and talk to us about the NRET project?
I'm Dan Daugaard. I'm a finance professor, so I'm one of those exciting people that people go to university to learn a lot about numbers and be put into a deep state of sleep and then come out and know how to run the world, run banks. But I have a friend here, and his name is Dr. Richard Ahadzie, and he's a giant, literally, obviously you can see, with numbers. And he and I work together on many different projects, and in this particular project, it's working on the dollar numbers around irrigation investment and the returns from that, as well as my part is more the finding the holes and challenges that then fold into subsequent research. So I can see Nigel looking at his watch already when I'm introducing. So Richard, do you want to take us through the big picture, and then I'll follow?
Thank you, everyone. So our project mostly focuses on trying to find the economic impact of irrigation to Tasmanian economy. And our research team has three main goals. And the first one is the microeconomic analysis. And basically this looks at trying to see the economic value from the past data that we have, which is the reports that we're able to collect. We're able to collect 47 reports, and these reports were on irrigation schemes and small farm schemes. That's report on the benefits of irrigations and the outputs. I would like to say today's presentation is consisting of both two teams, Team 1 and Team 2. With Team 2, we looked at the data from ABS data, and we tried to identify the agricultural output patterns and see how irrigation can, the importance of irrigation from the regional sectors. So we look at north, northwest, and south part. The last part of our team is yet in progress. We will start that next year, and we'll be looking to hear from all of you here. So feel free to talk to Dan and I, where we'll try to gather surveys, how we can impact on designing the surveys, and to see how we can collect data on that. So for irrigation in Tasmania, we know that the agricultural input in Tasmania has increased from 1.1 billion to 2.1 billion in 2022. No, sorry, 2021. We don't have data beyond 2021, sorry. But the Northwest has been a critical part of this growth. Northwest and North regions contribute significantly to the Tasmanian agricultural output. When we look at the Northwest case, it increased from 420 million to 860 million. And irrigation plays a key role in that. And this comes from the fact that dairy is contributing significantly, vegetables is contributing significantly. And this would not be possible without TI. And TI has schemes like Safran's, Wiley, and Daugaard, which are structured in a way to promote high-value crop production. And it has improved both the agricultural impact on the irrigation. For the microeconomic benefits, I will say, as a state, Tasmania is enjoying from irrigation. It is documented that for every $1 of public funds spent, there is around $3 economic benefit. Yes, this $3 is conservative. It will be more than that if we have the current results. And I'm happy to hear the minister said it's around $8. So there is progress in having irrigation on board in the state. For the dairy, one may ask, what are the factors helping the Northwest? One is dairy. Dairy is very important. It contributes significantly to the growth of irrigation of agricultural output. it increased around 290 million in 2021, with irrigation playing a key component of that. This is because the dairy industry relies on pivots and irrigation precision technologies, which are all relevant in having the outputs. Vegetable is part. Vegetable has grown from 90 million in 2010 to almost 200 in 2022. And this is also due to the fact that irrigation is implemented with TS schemes that provide water reliability around 95%. It's worth mentioning that other horticultural crops also benefit from irrigation, and this is possible due to TI schemes that we have. To give you a bit of graph, we have the states, as we can see, from 2010 increasing to about 2.4 billion, with the north and west part contributing significantly. And we also have the crops and specific products that contribute to the output. You can see a structural shift from 2016 to 2017, where dairy and vegetables and livestock products are driving the impact, with a shift away from wool and non-dairy products. Due to time, I'll leave it to Dan to continue from here.
Thanks, Richard. Hopefully you picked up from those graphs the incredible rise in revenue, agricultural revenue across the state, and quite subtly in the first diagram you would have seen the shape of the northwest is a huge spike, and then in the second table there's the reason for the spike, and that is of course dairy and vegetables, and it's this spike that has led to the statewide growth, significant growth in agricultural revenues. And so I want to tie that in a little bit with the irrigation schemes. And of course, we're aware of the reports that we were able to document and synthesize point to the dollar numbers that the dairy industry has benefited from the Circular Head irrigation scheme, but then in terms of Sassafras Wesleyvale supporting both the dairy and vegetables being integral to that success. And the reports that we've simulated, not just about the numbers, are also about surveyed responses of farmers to the presence and existence of the irrigation scheme. And so what that means is if it does cut the mustard in terms of the costs being palatable, both the irrigation costs as well as the fuel costs, to actually produce profitable shifts in choice of what you're going to grow. There's also this secondary benefit going on from farmers who engaged with that, and that was this increased confidence that in fact the water will be there when they need the water, if in fact there is a continual change in availability of water. And so the merits of the scheme in terms of both those industries have been both the turning on the tap as well as the actual safer trade-off in terms of risk of change. Now, in the Northwest, you represent the exemplar of change, and there's been behind the rollout of the irrigation schemes an incredible shift in terms of what we grow and what we're generating revenues and profitability from. And as an example of that, you would know more than I would that broadacre crops pretty much diminished significantly, dropped in terms of their contribution to revenues, and were substantially replaced by those numbers that you had were threefold and twofold increase in revenues from dairy and from vegetable. And so while that speaks to something that is actually happening across the rest of Tasmania, the transition to more profitable agricultural production, what is quite strange is the degree to which in the Northwest, in total irrigation volumes in water, we're actually decreasing. And in addition to that, we're decreasing the amount of land that's being irrigated. And so what's happening in the background is a significant amount of efficient use of the available water. Now, only a small proportion of it in the northwest is actually coming from the irrigation schemes. A large part is actually the rivers and your own saved waters, but that small proportion is actually growing year by year. And what it actually points to is the potential for capacity of growth, doing what we do in terms of efficiency, but actually expanding it to other activities. And you can see I'm carefully avoiding eye contact with Nigel as he holds up a 5-minute, 3-minute, and then a 1-minute sign. So very quickly, I just want to give you the sort of bad side of this, which creates problems that then TIA and other researchers are seeking to fill. And that is just some of the challenges that are stopping the ongoing efficient use of the water. And that is in terms of inefficiencies, particularly with scheduling, and the possibility that if you're not starting up at the right time to the right extent, you drop back in terms of your efficient eventual produce. But more than that, in terms of the soil health, the great variability is pointing to the need for greater innovation around how to use the available water. And slipping straight through to the big solution is we've had a great fortune of moving around the TIA and the state government workshops for farmers and seen two major things come out of that, echoed by the TIA research. And that is there's a growth in not just knowledge, but comfort, familiarity and trust in the biometric tools, digital tools to actually know where and when to apply the water, but as well as that, a greater sense of how to do the optimising, the trade-offs to work out whether I do afford or not the water. And the one other thing that came out of the workshops was the degree to which the simple coming together and being part of it, very much like this session through the rest of today, builds a considerable amount of community support from others in a similar position. Nigel's going to race up here and move me physically, so thank you very much for the opportunity of talking to you.
Don't step away, Dan and Richard. We do have the opportunity for a few questions. So if anyone does have any questions for Dan and Richard, please raise your hand or call out loud, and we do have a mic to roam around as well. Any questions for Dan or Richard on irrigation in Tasmania? There must be lots, surely. Do you want to come and do mine? Got one here.
Yeah, thank you. So you used the term irrigation dependency in your talk. Can you define what that means for us? Or for me in particular?
So our numbers are mainly focused on irrigation schemes, but out of that work we've been able to collate much of the TIA reports or experiments move beyond the use of schemes to actual all sorts of use of water, the water from the dams, the water from natural sources. So the insights in what's holding back efficient use of water, of irrigation, is quite general, whereas the dollar numbers for across the state and how it's dovetailing in with aggregate agricultural revenues are mainly the irrigation schemes, so it's TI's numbers. So the general lessons are everything, but the dollar numbers are TI.
Can I invite Jon to the stage to talk to us about insects in agriculture?
Good morning, everyone. Nice to be here. So I'm Jon Finch. I'm one of about 9 entomologists that works at TIA. That includes our PhD students as well. But we have quite a presence across the state now, and we try and get ourselves involved in all kinds of research across agriculture, because insects are involved in all parts of agriculture for good and for bad. And so I've got to kind of really, I feel like a little bit of a fraud here because I don't actually work on vegetables. Most of what I do is on fruits, so berries and apples and cherries and things like that. But what I thought I'd do here today is to come and talk to you about something that I think is really important and something that's going to be relevant to you guys. And also to kind of use this as an opportunity to get some feedback from you. So to really start this conversation and to kind of pitch these ideas to you and see what you think, and you can come and talk to us about it and say, yeah, maybe that would work, but maybe not. So this is an opportunity to engage in that conversation, and feel free to ask questions throughout. I'm happy to do that, or heckle as you like. But what I'm going to talk to you about is something I think is really important. So the problem, so you're all aware of this, and Mike's already touched on this already, is that one of the biggest threats to productivity in Tasmania and to agriculture across the country in general is going to be climate change. Climate change, well, what does that really mean? Well, we're talking about elevated temperatures, something between 1.5, although we've passed that now, and 4 degrees of temperature change over the next 50 to 100 years, depending on how we react to that. So that's going to be quite a lot. Bear in mind that, to put that in kind of context, if we think about the Ice Age, 10,000 years ago, that was a 5-degree drop in temperature. And that meant we had ice sheets coming all the way down from the North Pole to London. So a 5-degree drop means a huge change, and we're looking at going about 2 to 4 degrees in the opposite direction. So it's going to be pretty scary. So it also means elevated CO2 levels in the atmosphere. And it also means changing in precipitation and more extreme weather events as well. And the thing about insects is they're influenced by all of these things at the same time in quite complex ways. So I want to take some time really here to talk about that and how it's going to influence things in the future. So to quote the famous climatologist Nelly back in 2002, thanks for those of you who got the reference, it's getting hot in here, right? Things are starting to warm up. And insects are what we call cold-blooded in the layman's terms. You might want to use the posh term, which is poikilothermic, meaning that their biology is very much sensitive to rising temperatures. And basically what this means is when we increase the temperature, insects grow faster, they reproduce more, they have more generations per year. That's up to 5 additional generations per year for things like aphids, 5 additional generations. And it means that in places like Tasmania, where we generally have cold winters, they're likely to have improved survival over those milder winters as well. However, it's important to say that really hot temperatures can kill them off too, and it's quite complicated to work out exactly what the impact is going to be, and we almost have to go on a species-by-species basis. So that's temperature. The other kind of more interesting thing, I think, about the way that CO2 is going to affect insects is their impact on plants and photosynthesis. So interestingly, when we have more CO2 in the atmosphere, it actually enhances photosynthesis. Great, plants actually grow more when they get access to more carbon, when we're at whatever, 500 parts per million, which is what we're looking like we're probably going to get to, or something like that. So as a result, the relative amount in, so the plants actually photosynthesize more, which means there's more carbohydrates in those plants. And as a result, the actual amount of nitrogen in the plant, the relative amount of carbon and nitrogen begins to drop. The C to N ratio actually increases. And so what happens then is to compensate for that protein-poor food, herbivorous insects, pest insects, well, they just have to consume more leaf material to meet their metabolic needs, and this directly increases crop damage. So to kind of put this in a little analogy that I like to talk about here, this is Eddie the Beast Hall, former world's strongest man. And this, if you think about an athlete, if we use this analogy, then if we think about an athlete who's training for a big event, something like that, they're going to switch from a high-protein, steak-based diet to eating marshmallows, okay? So something that's way higher in carbohydrate. And although the total amount of calories consumed has actually increased, the athletes can't meet their nutritional protein requirements, so they just have to keep eating and eating more to get that necessary protein, and they consume, therefore, a much larger volume of food. And basically what that means, if we take that analogy and move it over to the insects, is they're going to be doing a lot more damage. The protein content of the plant is likely to drop something in the region of 15%, the nitrogen content, and the insects are likely to increase their consumption by about 20% to make up for that. So there's a lot of damage going to happen in the future. And how that works is going to vary between the different insect species, so it's a complex picture that I can't go into now, and it will vary between the different insects and pests that we're looking at. The other thing to make clear is obviously that drought, we've already talked about irrigation, so severe drought causes plant stress, and that makes some plants more susceptible to both pests and diseases. And we've talked about irrigation. Well, irrigation can be really good, but it can actually make crops more resilient, but also makes them more attractive to pests. What often happens, or seems to happen in Tasmania and Australia, is that a few months from now, the landscape starts drying off, and the only green and growing things in the landscape are those irrigated crops, which means the pests concentrate on those few resources in the environment, and that's something we need to be aware of. Heavy rainfall, well, not great for anyone really because of the erosion potential. Well, that can wash away small pests and things like that, but probably it does more harm than good. Unfortunately, when we have these swings between drought and deluge, this can favour pest populations, and that's because drought really knocks back our key predators. it hurts them much more than the pests, and they take longer to come back after that drought. So we have a drought, and then the rain comes, everything starts growing again. The predators potentially going to be left behind, and this basically means that our predators are not going to be able to keep up in the same way that they do now, which is something we have to be aware of. Likewise, as the temperature warms, what we're going to probably see is that pests are going to start shifting their range from where they are now towards the pole, so the South Pole or the North Pole. And that means that that's going to amplify both the frequency and the spread of some of these alien species that we might be worried about. So pests like Q-fly, which we had an incursion from a few years ago, and fall armyworm will be more capable of surviving through those more mild winters and causing a problem for us year-round. And that means that strong biosecurity is going to be more critical than ever in order to keep these things out for as long as possible and maintain productivity within the industry. Another important thing to understand about this is what I call missed opportunities. So warming can really disrupt the timing between pests and their natural enemies. So if temperature rises, so a way to think about this is, pests respond to temperature, we understand that. But what happens if the pests respond to temperature, as often happens, but maybe our predators, the things in the landscape that deal with them, they might actually be timing their activity to something like day length. Day length doesn't really change. Well, it changes throughout the year, but it's not going to change on an annual basis. And so if our pests are responding to increasing temperature, but our predators are responding to day length, they're going to come out of sync, which means the job that they're doing now in order to take care of our pest populations, they're not going to be doing anymore, and we're likely to see an increase in pest numbers. So what does all this mean? Well, in various studies that have looked at this, we're anticipating that crop losses due to insect pests of just 3 major crops, rice, maize and wheat, are going to projected to be increased by 10 to 25% per degree of warming. And we're already at 1.5. And we're looking at maybe getting to 2 or 3 by the end of the century. And what this means is that global crop production is expected to decrease by approximately 10% by the mid-21st century, of which we are now 25 years away. This, of course, is in the context of being asked to feed more and more people as we go forward, moving towards whatever it is, a population of 10 billion by the end of the century. So something we need to be very aware of. So, how do you think growers will respond to that, to increase pest pressure? Open question. So we had plant breeding, yeah, it's possible, although plants are not routinely bred for resistance to pests, they're bred for lots of other things. What? Yes, exactly, thank you very much. So it's very likely that the response to this is going to be more pesticide use. That kind of goes without saying. If people feel threatened, their livelihoods feel threatened, they're probably going to spray more things. And that brings us on to something that hopefully Hamney, wherever he is, recognises from this year. There we go. Hamney will be graduating next week, hopefully, but he probably saw this a few months ago in my lecture. So this is what we call the pesticide treadmill. It's an old analogy, but it works really well for how people respond to increasing pest pressure. We sort of changes in products available. So a pest comes along, we apply a new product, that pest is controlled, we continue to apply the same product, the pest becomes resistant, we apply even more product, it becomes even more resistant, and we apply a greater frequency until the pesticide no longer works. And so from that perspective, it just gets worse and worse. And that's why we can't afford to start doing that. And we need to think of new things to do. So what can we do? Well, we need to do more work in this area because strategies like crop rotations, intercropping, which I'll talk about, which is what we've got the picture of here, and also potentially regional production breaks are likely to become even more important for managing the growth of these insect populations going forward. However, we still kind of lack research that tells us how effective these are going to be long-term in Tasmania, and this is something I kind of am really interested in as a research opportunity going forward. So this is a study from a few years ago which looked at this kind of what are the benefits of intercropping, and by intercropping I mean growing things in strips, in rows. Instead of growing it in a big plot altogether, we grow things between each other to try and break up how much crop is available and how quickly things can move. And basically what the evidence seems to suggest is intercropping has really good benefits for managing pest populations by stopping their growth, but also has a range of other benefits like soil health, like soil fertility, water regulation, carbon sequestration, stuff like that. So the question I'm posing here really is, can it work here? Could it work? Would people try this? Is it feasible? And that's kind of what I want to get some feedback from you guys about. So yeah, that's my ulterior motive for here. And we also need better forecasting. We have really good models for forecasting crop growth. We have really good models for forecasting insect growth, but those models don't talk to each other, and they will influence each other. It's a complex system with feedback loops, And so we need to understand how those things work, and we need it probably to be automated to allow people to get on top of those things. So all this thing have to work together: better forecasting, better models, and automated modelling to really get on top of this going forward. So key takeaways for the end of this. So we're likely to see more pests and more damage under climate change. I'm sorry, that's just the way it is. And it's important that we don't fall back into those old habits, just spraying more to get rid of that problem. And cultural controls, monitoring, forecasting are all going to be really important to maintaining or even increasing our current levels of production going forward. And that's all I have to say. And I welcome any questions or chats later on. Thank you very much.
Thank you, John. Are there any questions for John?
Question around the forecasting and the modelling. Could we ever get back to regular sampling to see when flights are coming in from Australia or mainland?
Well, yeah. I think the question is, that was done for a long time, obviously. And I think the real thing is who pays for that, who does it, because it was done for a long time. Was, I think, before my time, was pretty useful, but I think there's no funding for that at the minute to do it. And so the question was, how useful would that be? And how do we start that again if it's really necessary?
How much will it cost?
Yeah, that too as well. I don't know the answer to that question, but these things are done routinely in other parts of the world. In the UK, they do it, and they have big air vacuum cleaners that sample the air constantly and suck things in and see what's flying around, and then use that as a forecasting model for growers. I think there's another question down here at the front.
Hi, thank you very much. I just have a question about that combination of the CN ratio changing and the irrigation, and therefore obviously we're putting nitrogen fertilisers on crops as well.
Yep.
Wouldn't we be bringing more of the pests out of the natural environment and into our farming and cropping areas?
Potentially.
Because of,
so
the combinations?
Yeah, so it's complex because you're putting crop physiology, which I'm not an expert on, and climate change and insect behaviour all on top of each other, and how they react might not be very predictable or not very well known. But so if the landscape dries off, that's going to slow down plant growth, and there's going to be this lush, fertile crop just waiting to be attacked.
Yep.
And so we might see in those conditions more activity.
So is there any research happening in buffer area plantings? I know you're looking at interrows, but also to have maybe a predator habitat around fields so that there's somewhere for them to stop and predators to increase, but pests
so that you're absolutely right, you can do that. What we're doing some research in the berry industry, potentially using what we call trap crops to try and intercept things as they come in, but it's still quite hard to do because you have to sacrifice some cropping area in order to do that, and then you have to really demonstrate the economic benefit that that's going to help the grower, and at the same time as growing a berry crop, then you also have to grow, for example, an alfalfa crop next door and keep it healthy enough so it increases the complexity. So we can try that, and we are doing that, but we still don't have the data to say, that'll definitely work yet.
Thank you. Not sure if I want to open this can of worms or not, but just in terms of looking into the future and gene editing and GMOs, does that play a role in protecting crops or protecting fruit in your case?
Not in Tasmania. Obviously we can't have GMOs here. Obviously they're used very widely on the mainland and have been very successful in for example in cotton. They've been used really effectively to manage pests in that for a very long period of time. But could it be part of the answer? Yes, definitely. Could it enhance our ability to respond to things? Yep, absolutely. But as you know, it's a legal thing, isn't it? It's beyond my ability to forecast as a scientist.
Thank you.
All right, thank you very much, John. Can I invite Tamieka to the stage to come and talk to us about her pyrethrum research?
So good morning, everyone. My name is Tamieka, and I'm part of the TIA team that work here on the Northwest Coast. And this project is funded by the state government's Agricultural Development Fund, as well as Botanical Resources Australia, who grow all of Australia's pyrethrum. And the project aimed to try and evaluate a more rapid method of detecting pyrethrum pathogens for disease management. So BRA are a global leader in pyrethrum production, and it's grown to harvest the seed, which has a high concentration of pyrethrins. And pyrethrins are chemicals that have insecticidal properties. They're extracted from the seed locally here in Ulverston and then formulated into commercial pest products. One of the factors that decreases pyrethrum yield are fungal diseases. And BRA use an integrated approach, including things like clean seed, site selection, and fungicide programs to minimise disease losses. And TIA have worked with BRA over the last 25 or so years in that disease space. We've looked at what species infect pyrethrum, we've looked at their biology, we've looked at the efficacy and timing of fungicide programs. But two of the pathogens that infect pyrethrum are the target of this study, and they are tan spot, which is caused by Didymaella tannacidi, and ray blight disease, which is caused by Staganospropsis tannacidi, or is also called FOMA for those who've been in the industry for a long time. Now, while ray blight shows these shepherd's hook characteristic on our flowering stems, the disease symptoms on leaves throughout winter and spring are indistinguishable in the field. So we can't go out into a paddock and say that paddock has a lot of tan spot or that paddock has a lot of ray blight. The research that TIA have done over the years has shown that if we can decrease early season disease, we decrease late season disease, and we have an increase in yield. We've also shown that the two pathogens have a differential response to the fungicides that are available for their control. So being able to determine the amount of disease that is in a site or a field could be really valuable for management in terms of identifying what fungicides we might apply or how many need to be applied to the field. And the way that TIA's R&D team and BRA's R&D team identify the pathogens in the leaves is through a process called leaf isolations. And in this, we take leaves from the field, we take a small piece of tissue that has a symptom, a couple of millimetres squared, and we plate it out onto a media. That then gets incubated, which gives the time for the pathogen to grow out, and we identify it based on its morphology. And while this process works really well for R&D, it does have some limitations in being able to identify disease in paddocks over time. The first is it requires expertise. You need to be able to tell the difference between the different pathogens and what they look like. It's also quite labour intensive, and you can only process so many leaves per day, meaning you can't really apply it to every crop that's in the paddock at a time. The other issue is it only looks at a very small piece of leaf tissue and only tells us if the pathogen is present or absent. And the results aren't immediate. There's a lag period between when we do the test and we get the results. And in that time, the pathogens probably become more established in the field. So what this project has decided to look at is a molecular approach. And in a molecular approach, we're trying to detect the pathogen DNA in the plant tissue. And for this, we take our leaf piece or a selection of leaves, we extract the DNA. We then run a test that makes copies of the target DNA, so the pathogen DNA that's in there, and from that we can get a result that either is a negative or positive, that the pathogen is there or not, or we can quantify the amount of pathogen that's in the sample. So when we started this project, we came up with this overall project approach, which combines the three components of leaf sampling, DNA extraction, and some sort of assay to develop a test that is specific to the two pathogens, that is sensitive enough to detect them at low levels, is cost effective and fast. And as we went through these, we came up with all these other questions that we needed to answer to be able to develop this test. We also realised that all of these aspects were connected and linked to each other, and we had to work on them concurrently. And an example of that is that the type of assay we use will determine how good a quality the DNA extraction needs to be, or the amount of leaf tissue we want to use will determine what sort of DNA extraction we can use. The other thing we were really aware of as we did this project is that we were developing something for industry adoption. If we were doing something just for our in-house R&D, we would probably take a completely different approach. But we realised we needed to have something that we could validate against the isolations we'd already done. It had to have a benefit that the industry didn't already have. It had to be economical and give a benefit to the farmers relative to the cost of what the test cost. We spoke with the industry a lot throughout this project about how they would best apply it. And at the moment, we're looking at using it to try and decrease the number of fungicides that might be applied in the field, which would give a significant economic benefit to the farmers. And then we also had to make sure the industry had capacity to use it. There was no point developing something that they didn't have the equipment or the expertise to use. The first thing we focused on was the assay, because if you don't have an assay, then the rest doesn't really matter. And so to help us with the assay, we did genome sequencing of the two pathogens to give us more data to develop our assays on. And we also sourced DNA from different pathogens that infect pyrethrum so we could make sure that the test was specific. And while we looked at a whole range of different assays and many got thrown out because they weren't economical, we landed with 3 that we'd have to use. I won't go into too much detail because we're short on time, but the 3 we have is standard PCR, which is on the left-hand side. Standard PCR, we have to run a separate test for Didymella and a separate test for stag, and we get a positive or negative result based on if we can see a band on a gel. The middle one is a lamp assay, again a separate test for Didymella and stag, but this is more simplified, and it was chosen because of its simplicity. It just needs to be incubated at 65 degrees. It's really cool in that as this reaction happens, if the pathogen is present and copies of the DNA are made, it releases hydrogen ions into the solution, which changes the pH, and we can identify that pH change with a colour indicator. The final one we've got is qPCR, or quantitative PCR. This one allows us to test both Didymella and stag in the same tube and allows us to work out how many copies of the pathogen are present. And it can detect down to 10 copies in that test. So this is the test, the qPCR, that we've decided to run with in terms of looking at how much disease is in a field. The next thing we had to talk about was how we're going to sample leaves. And initially in this project, we started with very small pieces of leaf to validate that we could actually detect the pathogens. But as we moved on to thinking about how we would sample fields, we had to have a think about how we would sample leaves. And we kind of went back to basics, that the total amount of disease, which is what we want to measure, is a combination of the incidence, so how many leaves are infected, by the severity, or how infected they are. And on the left-hand side there's just an example of 3 different samples of 3 leaves with different levels of incidence and severity, depending on how we would measure them. And when we tested this in the lab, we took leaves we'd infected in the lab and mixed them together in different ratios. We found that leaves that had a higher incidence level didn't necessarily always have a higher amount of pathogen. And we took this one step further in that we took leaves from the field that had multiple symptomatic areas, and we did multiple isolations from each of the spots. So from a total of 12 spots per leaf, we were able to identify how many different pathogens were present. And we got some really interesting results. So we found that in the 20 leaves we looked at in this particular paddock, there were 2 to 5 different pathogens associated with each leaf. For some of those leaves, we had both Didymella and stag present. For some leaves, 1 spot gave Didymella, and for some leaves, 8 spots gave Didymella. So there was a huge range in both the difference in incidence and severity on those leaves. And so if we'd just done isolations, and it would depend on which spot we'd picked, we'd get a very different result. So it kind of made it really clear that we needed to be careful about how we would pick those leaves. The final piece of the puzzle is the DNA extraction. And the DNA extraction is probably the most time-consuming and expensive part of the whole test. We trialled many different tests and types of extractions. We used commercial kits, microneedle patches, one-step extraction buffer, you name it, we've tried it. The issue we came across is that commercial DNA extraction kits, which give us really high-quality DNA, are also very expensive, and they will only take up to 100 milligrams of tissue. And if we want to sample fields, 25 leaves is about 10 grams. So the commercial kits weren't really able to do what we needed them to do. So we've come up with a trial that we're currently testing in our lab, and it's got 2 different approaches. The first is we grind our leaves in liquid nitrogen to disrupt all the cells. The second is we use Ahadzie extraction bags to disrupt the cells. I don't know if you've seen them, but they're like a mesh bag with a mesh lining, and you put your leaf samples in with a buffer, and then you rub it really hard, and it disrupts all the surface tissue and releases all the pathogen DNA into the buffer. From that, we're looking at whether we can subsample that mass amount of leaves and do a DNA extraction, or we can develop our own in-house bulk DNA extraction protocol. So far, we've been using the pie paddock here at Forthside, which you'll get to see later on, to test how well this works. And what we've found is that even though there's no stag in that paddock, which is not good for us, but is good for the industry, it did have a level of Didymella at 15% incidence, and our qPCR test can pick that up really, really easy. So we're getting some good positive results. So what's next? The next step for us is to complete that DNA extraction development and make sure we have a really good test that we can use consistently. But next year we're going to do a whole lot of field sampling. And so that will be sampling bulk leaves from fields and performing our DNA extraction in our assay to look at how the amount of pathogen we can detect with a molecular test compares to what we see with isolations. And TIA have done enough isolation data over the last 25 years that we know what the isolation data looks like of those two pathogens over the season. So it'll be really interesting to compare the results. It'll also give us a really good baseline data on what we expect when we use molecular testing. So from those results, hopefully we'll be able to see if we see variation in pathogen incidence and DNA detection in between our different fungicide programs. And we will go from there to see what happens next. Massive thank you to our project team, particularly Stacey Pilkington, who is our amazing technical officer, who's filled many, many lab books with trial data for this project work, and to our funders for continuing to support us.
I'll invite Tory to the front to come and talk to us about the net zero emissions trial.
Thanks, Nige. So I'm Tory, like Tamieka, I'm based up here in Burnie in the northwest. And I'm the last talk before morning tea, and I'm not going to talk long because I'm going to invite you out into the field to have a look at the trial that we're running. But I just wanted to give a little overview and kind of explain what we're doing and why here while it's easy to hear me, and then we'll head out and we'll show you in the field what we're doing. So this project funded by NRE Tas, the Tasmanian government, and it's a 4-year project that's really focused around understanding nitrous oxide emissions in horticultural systems. And we're looking at vegetable systems, so trying to understand what the emissions are across the rotation, so as we move from different cash crops and through fallow periods, what actually happens with the nitrous oxide emissions from those systems, and also looking at opportunities to mitigate and lower those emissions. So why do we care about nitrous oxide emissions? We've talked a little bit today about the climate impacts, and we know that emissions contribute to this global warming effect. And nitrous oxide is a really potent greenhouse gas. It has a much higher warming potential, up to 30 times of what carbon dioxide is in the atmosphere. But I think what's also really important to point out is that nitrous oxide emissions, what they actually represent is nitrogen lost from the system. So when you're growing something and you're adding nitrogen fertiliser, a lot of that goes into the plant, that's what we want, and emissions are actually nitrogen that hasn't been utilised by the plant. So as well as this environmental aspect, there's also an opportunity here to actually increase the nitrogen use efficiency in our plants, capture more of that nitrogen where we want it in the plant to build protein, and by doing that, we're actually going to lower the emissions. Emissions, nitrous oxide emissions, it's not an even linear production, so it actually happens in pulses or spikes, and they're really heavily influenced by the management. So when we add nitrogen to the system, this can be a trigger for a nitrous oxide pulse, and also events like rainfall can also trigger emissions. There have been several tools developed, which you might have heard of, around estimating what your emissions are. And so these are kind of like an online calculator, like a spreadsheet. You put in what the crop is that you're growing, when you planted it, how much fertiliser, a heap of other information, and it spits out a number for you to tell you what your equivalent tonnes of carbon dioxide emissions are. We don't have a lot of validated data for Tasmanian growing systems. We're in a cool climate here. We've got different processes, different effects on the processes because of that temperature and our rainfall events that we have here. And so this project is really about trying to validate what are the actual emissions produced in our systems here. Because if we're going to have a target or a way to move forward to lower the emissions, we actually need to know what they are first so that we can measure that and quantify what are we actually producing, do we need to lower it, and can we measure the effect of mitigation strategies against that. And the other aspect that I want to highlight here is any mitigation strategies that we want to use, they need to be economically sustainable. So if you want to have an environmentally sustainable production system, it also needs to be economically sustainable. We can't afford to spend more than we make growing veggies. So background on nitrous oxide emissions, when we add fertiliser, that's adding nitrogen into the soil nitrogen pool. And there's kind of 3 pathways of that nitrogen. So where we want it to go is into the plant to be used for building the plant and making protein. It can also be lost through leaching processes, so this is coming out into the groundwater, and then it can be lost via emissions like nitrous oxide. And we already have a really good understanding of what mitigation strategies are available already that can help reduce these emission losses. And so a really key one is around our nitrogen application timing. So really optimising this. We're giving the plant the nitrogen when it needs it, and we're not leaving excess nitrogen in the soil to then be converted and transformed into gas emissions or leached out in the water. Other factors that can reduce emissions are things that minimise soil disturbance. So we know when we turn the soil over, that can trigger these pulse events. And things like having a cover crop in after your cash crop can be beneficial. Part of that can be it can actually mop up any leftover nitrogen that you've left in the soil. But if you use legumes as part of that cover crop as well, it can actually add some nitrogen back in so that you don't need to put as much fertiliser on for the next cash crop. So then you're reducing that fertiliser input. There are also new technologies being developed around the fertilisers. And so fertilisers that have coatings or contain chemicals that actually inhibit some of these transformation processes. So nitrification inhibitors, and that can help lower the emissions produced during your cropping cycle. But as I mentioned, if we're going to try and reduce emissions, they need to be practical and achievable for growers. They need to be economically viable, and importantly, they need to work. They need to lower the emissions like we think they will. So we're running this trial here, firstly to validate and baseline what are our emissions that we're producing, and then also to have a look at some of these mitigation strategies and can we actually see, are they commercially relevant, are they cost effective, and do they work? So it's a 4-year project, and we're running a 4-year vegetable rotation, so going through potatoes into broccoli, carrots and onion. We're going to head out in a minute and I'll show you this in the field. But every year we will grow each crop and then we will rotate through the block. So we're going to end up with a lot of data across the 4-year cycle, and this allows us to really separate out different environmental effects that you might see seasonally with what's happening in the crop. The other side of this slide, I'm just showing the bed example layout for one of these sub-blocks in our field. So this would just be, say, for onions. And we've divided that into 4 replicates so we can do some statistical analyses on the data that we get. And we're comparing 2 different treatments here. So the blue beds are showing conventional management. And so for each of these vegetable crops, we've worked with industry representatives on our steering committee to develop kind of a representative management program of what typical nitrogen inputs would be for growing that crop commercially. And so they're the management processes and the inputs that we'll use under our blue beds there and the conventional management. And then again, working with industry, we've looked at what mitigation strategies could we actually implement that are ready to go right now and practical for growers to use. And we've built a program of trying to reduce fertiliser inputs and complementing that through other strategies to try and minimise emissions. So we will be measuring the emissions directly in the field. So we have some specialised chambers that we use in the field that actually collect the gas from the soil. And then alongside the emissions, we're also going to be measuring a suite of other data points around the soil and the plant growth, and that really helps us to understand why these emissions are occurring. So we'll know how much emissions are happening and also understand what's driving them. So we're looking at the soil content, the nutrition in the soil, the structure of the soil moisture. We know all of these factors are involved in those microbial processes that drive emissions. We'll also be looking on the plant side about how the plant is actually using the nitrogen that we apply. So how is that nitrogen being utilised in the plant and how is that affecting its growth and performance? At the end of the season, we'll have yield data from our trials as well as quality assessments. And then what we'll do is we'll take all of that around our yields and our performance and the emissions data, and alongside that, working with economists at NRE Tas, we'll actually do a return on investment analysis. So how much did we spend to grow that crop? What did these mitigation alternative management strategies actually cost us in terms of dollars? And what is our net return at the end of that? I also wanted to take a moment just to highlight as well that this trial, we're calling it a demonstration trial, because while we're doing a lot of really exciting research and technical data collection here, it was really important that we have something that's actually accessible for people to come and see. How do we measure these emissions? What does it mean? What does it look like in practice? And as part of the project, we'll be running workshops here at the farm to cover off different aspects of this so that people can learn about what emissions are and why they might want to understand and applications on their own farms. And then moving into years 3 and 4 in the trial will be supporting some grower-led trials. So if you come and you see a mitigation strategy that you think, actually, I could implement that on my farm, we're going to support you to take that, do the trial on your own farm, see how it actually works in your systems, because everyone's different. And if you want something to work, you really need to test it in your own system first. And then all of the data that we get out of this project, this is going to go into really supporting those decision-making tools. So things like those calculators that estimate your emissions, we can actually plug in our Tasmanian relevant data into that, so that in the future, when you're trying to estimate and monitor what your emissions are, you'll know that you can really trust the data that you're getting. So now I would like to invite everyone to join me and the research team out in the farm. It's just the next paddock over. So grab your hats and we'll walk out and I'll introduce you to the rest of the team and talk a little bit more about what we're doing.
Hello everyone, I won't take up too much of our time seeing as we're running into morning tea. But you can see here we have our 4 trial plots. So directly behind me here we've got our peas. Further down from that we've got our potatoes that went in about 2 weeks ago. In the far bottom corner there I've got my onions, and at the top here we've got our carrots. As you can visually see in our bed design, we've got our 3 different sample lots, and you can actually see, especially in the peas, the difference in treatments that we've got. So these ones here, the taller ones, they're our conventionally treated ones with working with industry leads, and then we have our alternative treatments, which we're going to start sampling those this week to look at the soil and the plant nutrients to see what they might need. If you look further down the paddocks, you can see that we've got these little pink flags. So they've got our chambers. So they're what we're going to be using to measure our nitrous oxide emissions. So we've got these little white collars here, and they get buried down into the ground till about our little clip. And what they do is they sit in there and they'll be there for the duration of the trial, and we'll sample during both with crop and in fallow. They're designed so that the plant's roots can grow in and around and not disrupt anything. And then what we're going to do on about a weekly basis is measure our nitrous oxide emissions. So what we do is, you can see Tory's got her little lid for our chamber there. They've got the reflective coating on the outside to help reflect any heat so we don't cook the soil inside. They get clipped on and they get left on for a period of an hour. And over that hour, we're going to take 4 measurements, and that helps us calculate the rate of emissions rather than just spot testing. So to actually measure the gas that's inside, you'll see on the top of the chamber, they've got a little round septum there. So we'll use our specialised syringes and some big long needles that I didn't bring with me today to take out a sample of gas. They'll go into a little vacuum container and we'll send them off to the lab for analysis. So that's what we'll be doing. Like Tory said, over the length of the trial, we're getting a lot of data. There's about 4,000 samples for me to take per year, so that's going to keep me very busy, but we're really looking forward to getting that data set to baseline what we've got happening here.
So Doug is our Forthside farm manager. He's been here for 6 or 7 years now, and very much the mainstay of what we do here at Forthside, and keeps Anne-Marie and Catherine in line. And we very much appreciate his contribution, his knowledge, particularly to Tas Seed and understanding of all things potatoes. So Doug, welcome to the stage. Look forward to hearing an update from you.
What I'm going to do, primarily it's on mop-top. We're only going to spend sort of 4 or 5 minutes on mop-top, but just do a rundown of Tas Seed and also PVY, NTN, where we're at with that. So I'm just going to rally through this really quick. Tas Seed is a not-for-profit, and we're part of, and T is part of a joint venture agreement between UTAS and the Tasmanian State Government. We're funded in full by seed growers. So seed growers pay that cost. Nothing comes out of TIA funds or UTAS funds or government funds for that. And as manager, I'm ultimately responsible to the buyer of the seed. A bit of a breakdown of what's happened over previous seasons. So we went through a low 23-24, 685 hectares. Last year was 810. The seed business has been growing. It's been absolutely awesome. A huge amount of confidence in industry, particularly seed growers, in growing quality seed and exporting that to the mainland. And that was up until the end of July when mop-top hit. it costs around sort of that $450 a hectare. The more hectares we have, the more efficient we come, so we actually lower the price for seed growers. If we do less area, seed price, the certification price goes up. There was a national standard for certification of seed potatoes. AusVeg were the holder of that standard. They said in May 24 that they no longer wish to hold that, which caused us a couple of issues. There's a certification scheme in WA, Victoria, New South Wales, South Australia called AusPicker, and then Tas Seed. So what we did, we formed our own Tasmanian standard so we can work from that. Got some help from some locals, built that, and it's working a treat. So to enable certification, we do paddock inspections. So the growers fill out a rego form at the start of the season. We do a minimum of 3 paddock inspections. So first one's about 5-6 weeks, aimed at any breakdown, so black leg or pecto. The second one at flowering to make sure that we have no mixes and the crop health is high. And the third one prior to desiccation. All G2 crops and some random later generation to actually test the tubers in the lab, and we look for Dickia, Dianthocola, multiple pectobacterium, but also a range of virus as well. That's just a copy of our field inspection report. Always nice to take a couple of happy snaps for later reference. Tuber inspections. So when potatoes are harvested, they're brought into cool stores. So 4 cool stores along the coast. And per semi-trailer load, 400 randomly taken tubers are extracted from that load, and we look at each and every tuber out of that. So a fair bit of work goes into that. We record that data either in our own system or Cherry Hills, for example, or Simplots, and we can give some pretty quick turnaround time. So when seed growers are harvesting those potatoes, they can actually make some changes to their grading practices if they need to. There's only 2 results from certification. It's either certified or rejected seed. And it's as simple as that. Others within the seed system may choose to buy that seed, so the likes of Simplot or McCain's will still purchase seed from the seed grower, and that may be outside of the certification standards, and that's classed as provisional seed. A lot of commercial growers haven't understood that for a period of time, but I think knowledge around that is now increasing. The mighty mop-top. So in late it was early July, we had an industry person playing around with some potatoes and realised something was astray, wasn't quite right with this batch of potatoes. So they did the right thing. And to that person, I say thank you very much. To actually put your hand up and ask the question is why, or what are we actually looking at? And without that person doing that, we'd probably still be where we were in early July, bobbing along as an industry. And we would not have the opportunity to address mop-top. Information is important, and I can't stress that enough. So with information, you actually act on it. If you don't have that information or data, obviously you can't progress. So Biosecurity Tas started a full biosecurity response with full protocols. Alice was appointed incident manager. Susanna was the state plant health officer. I think that's right. And both have been absolutely awesome to work alongside and work with. Not many people in state government actually understand potatoes, and they understood that very early, so they asked questions, and a lot of questions, which again, without those individuals asking those questions, we can't progress. So Bonnie Keller and myself were appointed as industry liaison officers to work alongside BT and to give them as much information as we can when they required it and to answer those questions as the process began. BT did some of our own in-house testing and yeah, it's small scale, but they're actually looking for it. So they dissect the potato, they're looking for any discoloration within that tuber and actively looking for mop-top, which is awesome. But it was soon apparent that that level and scale of testing was not going to meet industry needs. So with discussions with Biosecurity Tas and Intertech, so Tas Seed use Intertech historically for broader scale testing of our G2 program that I spoke on earlier. So we already had the system in place. It was only scale up of that system. So that's probably the largest area where TIA was able to help with the response in getting in the casual labour, organise the testing of those tubers, and progressing from there. hats off to the cool stores. So access to cool stores in July and August is really difficult. There are some lines that have been pulled out ready to cut, but the bulk of them are still tucked up in winter mode, all stored. So it's a large impost on cool stores at the time to ask them to pull out individual lines so we can actually grab samples out of those and then bring them back here to Forthside to get them carved up and sampled. Biosecurity Tas identified lines of concerns as we went, and they followed that through, again, trying to accurately determine the presence of mop-top within those lines. All up, Tas Seed tested 337 lines of seed or tests were conducted, which is a large number. So to generate a sample, I've got some photos here later on, but you take the stem end of the potato, you carve off the end of that, the very tip of it, you dig in with a little device and take a little core sample out of that. You put that in a group of 20, then do another 19 groups, so it's a 400 tuber sample, and we send that away and they test that. So after all that work with both Biosecurity Tas and Tas Seed, we determined that there is a very low presence of mop-top in Tasmania. It is not eradical. The way the virus houses itself within the soil and within the spores of powdery scab, means it's very hard to control, or you just can't. Important to note that there is a national mop-top working group formed a couple of weeks ago, and they're going to deal with issues such as standardised testing, which can't happen soon enough, but also around issues of movement of potatoes and what seed movement and general potato movement may look like in the future. If you've got any questions around mop-top, you want to go and have a look, the NRE website is a fantastic resource. So they've got some stuff on there for contractors, growers, if you're handling seed at cool store, that's your one-stop shop. So this is how we set the veg shed up. In the green tubs at the back of the shed there, they're the 400 tuber samples. We then pull them forward to the table, separate them out, do a count of 20 tubers in each of the blue tubs, carve them up, and then we pack them up and send them away to South Australia. That's just a photo of powdery scab, of which the mop-top lesions will house themselves within. Slideshow. So this is what mop-top looks like internally. You get that rainbow type effect through that tuber itself. The main concern is for the processing industry as such. So the photo on the left-hand side of the screen, if mop-top is in the tip of that potato, that will fry up differently. So when you go and buy that French fry, for example, from McDonald's or KFC, it will have a different end on it. So not the best outcome. It will lead to, rejections at factory. At the moment we're genuinely not seeing that at any real scale, so that's a good outcome. I'll touch on generations. So tissue culture is held within a lab, and then from that tissue culture gets out of tissue culture, it gets carved up and then planted, usually in a potting mix type material, or can be done hydroponically. That then grows out to be a wee little small mini tuber. So that's what seed growers purchase in Tasmania. And there's three main mini tuber production facilities in Australia: Ceylon in South Australia, Talangi in Victoria, and then Simplot based just down the road. Seed growers buy in those mini tubers, grow them out for the first year to produce generation 1 seed, and then over the future years, G2, G3, G4. So in Tasmania, we use primarily G3 or G4 seed to produce that final harvested product for factory. Simplot are moving that to a G2 planted system, I believe, which is a fantastic outcome. The reason for that is it's less opportunity for any virus, pest or disease to breed up within those seed lines. So, overall, internationally, we rate very, very well with that. Even at planted as G4, most countries plant G7, G8, G9 seed, which gives them more of an opportunity to house pest and disease within that. There's a photo of some mini tubers. Unfortunately, they made their way past biosecurity, which is not the best outcome. This was a couple of years ago. So the reason why that's not a great outcome is the stem length within that. So that will house pest and disease and virus. So something we've got to be mindful Just while I'm talking about viruses, PVY, the necrotic strain, NTN, was detected 2021. Thought we dealt with it pretty well at the time, then popped up again in 2023. So some of that seed had made its way commercially out into paddocks. There was 15 paddocks in total. And within that, industry determined we're going to try and eliminate PVY, NTN. So that's what we're trying to do at the moment. So out of those 15 paddocks that seed have been planted into, we have currently 8 paddocks that show regrowth potatoes. So the aim is to show 0 potatoes over the course of years. So management techniques such as livestock, chemical application over the top of crop or pasture, whatever that looks like, it's war on potatoes within those paddocks because we don't want any carryover of PVY NTN. Important to note that NTN is more of an industry concern and potential loss to potatoes than what mop-top is. So within those efforts, we've actually got a prohibited pest status that we're working towards with BT. And when IR9 is hopefully released sooner rather than later, yeah, that'll be fully detailed in that. That's what PVY NTN will look like. You end up with a really paper-thin potato skin in certain areas of your potato tuber. What's next? What's going to come into the state next that we don't know about? We've got neighbours in Victoria that are now have a tomato potato. That's spelled incorrectly. Good on you, Douglas. And TPP is now in Victoria and it is not controlled. What we have in Australia at the moment is there's been no detected indication of CLSO, and that is the bacteria that causes zebra chip disease. So we can have the psyllid without the bacteria, happy days. As soon as that bacteria finds its way into Australia, which hopefully it doesn't, but it could do, then we're going to end up with major issues. Bacterial wilt, are we going to have that pop up? Pictobacterium species, we are seeing those occasionally through seed lines. We've done a whole range of testing on that, but we just don't have, it's primarily cash, to work our way through what it actually means with those test results. PCN may come in, tobacco rattle virus. So biosecurity of orders is very, very important to us. Emma and Catherine, they're not in the room. They'll be over there in the kitchen doing their thing. For Tas Seed, these ladies are absolutely fantastic. The relationship with growers, it's second to none. No. Thank you very much for what they do. Any questions?
Thank you very much, Doug. We actually will call you back when it comes time for the panel discussion. There'll be a chance to ask Doug some questions then. I think it's really clear Doug's passion for the Tasmanian seed potato industry, and I think the industry's been really grateful for the amount of time and the amount of work that Doug puts in to industry behind the scenes. Many of us don't see the conversations and the phone calls and the discussions he has to represent industry, and I think we can be really grateful not only as farm manager here, but also as his management of the seed potato scheme. Can I call Calum to the front now to talk to us about new research for management of mop-top? Calum, over to you.
You may have, you would have heard this morning the Minister mention some funding for to do some work with mop-top virus. Well, that project is about to start, so I'm not going to actually say too much about that because we haven't actually got results as yet. But I want to talk a little bit more about mop-top itself and what is going to be done in relation to some of this work. This image here is what started it all. So as Doug mentioned, there was someone who was able to recognise something that wasn't quite right, and that was the start of our mop-top incursion. So relatively recently found in Tasmania, and powdery scab, which we have plenty of, is the vector which becomes problematic. So we've got plenty of people know the potatoes in here. Tell me, what is A? None of these are pathogenic. So these are internal damage to potatoes, and these are all physiological. So A is a bruised potato. B is internal cracking with growth. D is lack of oxygen. Oh no, sorry, that's a temperature one. So black heart, and that's a sort of a physiological fleck too. So we do have these internal defects. These are something people deal with. These ones are all viral. We have or have had all of these in Tasmania. Tell me, which one's mop-top? Anyone want to guess? I heard an A, I heard a C. It is A. A is mop-top virus. B, that's PVY-NTN that Doug mentioned that we have had and hopefully we are getting rid of. D is tomato spotted wilt virus, which is endemic in Australia. It's certainly present here. We have had entire crops wiped out. It's very sporadic. You know, once in 20 years it'll turn up and do nasty things and then disappear. Obviously it can make a horrible mess. C is one of those on Doug's list that is certainly of quarantine concern. That's tobacco rattle virus. It is in Tasmania, not in potatoes though. There is a record of tobacco rattle virus in daffodils, which shows another way of actually these things arriving into the state, so imported obviously at some point in the bulbs. What we don't have with tobacco rattle is the nematode that spreads it, which is probably why it's never hit into potatoes. If that arrives, potentially we have an issue. And there's another one. Again, in Tasmania, certainly not of quarantine concern, but again, can cause these necrotic symptoms. That's in a russet Burbank. Anyone got an idea what this one could be? Everyone's too shy. That's leaf roll virus that occasionally will cause necrotic symptoms in Burbank. And indeed, that's kind of why the West Australian industry gave up on trying to grow processing potatoes, because they got way too much leaf roll and they got way too much of this internal necrosis. So what does that mean? It means that mop-top can occasionally cause these internal necrotic marks, but we have other things that can do that too. We have physiological things, we have other viruses that occasionally do this. So we've been living with some of these for some time. So anyway, let's go back. So mop-top recently detected. It's carried by powdery scab, the pathogen, and this makes it particularly difficult to manage because powdery scab, the pathogen lives in the soil and can survive in the soil for an extended period of time. It can remain dormant in the soil for decades. The virus is picked up by the powdery scab pathogen from an infected plant and resides inside those resting spores, those little brown lumpy things, and it can sit in there and wait till a potato crop comes. So this is why it's non-eradicable. Certified seed is really important. Keeping our seed sources clean is really important, but we can't effectively manage what's in the soil. So a little bit about how it works. As I said, the powdery scab pathogen picks up mop-top from an infected plant. So if you've got a mop-top infected plant, it gets infected with powdery scab. The powdery scab pathogen will pick up the virus. It then, when it forms these spores, it goes either onto the tubers or into the soil and sits there. The way it transmits is, again, when the powdery scab pathogen wakes up, germinates, it has these little motile spores. The virus is sitting inside that spore. It swims through the soil, bites onto a potato root. The virus gets inside and away it goes and infects your potato plant. Foliar symptoms are rare. Now there's been a whole bunch of media out there and sites showing these horrible chevron-shaped chlorosis on potato plants. I don't believe there's been a single plant seen like that in Tasmania. There's certainly never been one of those seen in New Zealand, where this has been here for a couple of years. That symptomology appears to be related to what I suspect is related to environmental conditions. Where it has been seen is sort of like far North Europe, Norway, Scotland, sort of areas, and possibly related to day length and temperature in terms of that expression and that where you do get significant yield impacts. So we really don't know what yield impact there is, and certainly the New Zealand industry pretty well ignore this as not a particular significant issue for them. How's it spread? It's spread obviously in seed tubers. So again, cannot emphasise enough the importance of seed certification and ensuring that your seed is clean. Now that can be with or without any symptoms of powdery scab, because the virus lives, once the plant's infected, the virus doesn't need a powdery scab pathogen within the plant, and of course can spread to the tubers and spread that way too. In the soil, it's always associated with powdery scab, and obviously if you've got virus carrying or virulifera's powdery scab spores, anything that moves that around will move the virus as well. So soil machinery, contractors moving from farm to farm, wind and water, anything that moves soil around. What don't we know about it? We really don't have a good idea of distribution, and indeed that's what this new project will partly do, is to take some soil samples around the state and test those. We don't have any idea of impact on productivity, and we don't have really good tools for vector management. So what do we got? This money from NRE will look at these aspects. So try and get a better understanding of what we know about it or what we don't know about it. But it will look at detecting virus-infested soil. So providing a tool for growers so they can take soil samples, a bit like Predictor PT, test for Spongospora, test for our powdery scab, but also test for mop-top virus. And then determine risk, so you can make your own decisions in terms of risk of planting into that field. We're also really interested in tuber expression. Obviously this necrosis is important for processing. There is an indication that that potentially could be affected by temperature and therefore by cold storage. So we're going to be doing some work to look at how symptom expression might be affected by cold storage, and of course industry awareness. And lastly, from a vector management perspective, we have a Hort Innovation grant that's really looking at integrated management of soil-borne diseases, and this will focus a fair significant amount of effort on management of powdery scab and looking at new and novel control options, which will also directly or indirectly affect mop-top. Thank you.
Thank you, Calum. Calum, just remain here and there'll be an opportunity to ask Calum some questions in a moment. But can I also invite Doug back to the stage and Alice Clayton from Biosecurity Tasmania and also Craig Elliott from TIA. So Alice Clayton is the Biosecurity Engagement Management Program Manager for BT. And Alice, I've got some questions for you. And Craig Elliott is the Biosecurity Engagement Lead for TIA.
At BT, we've got a key role in preventing those pests and diseases from entering the state and establishing, that's all the border surveillance activities that you see in the surveillance. Everyone's familiar with the detector dogs. But what my team are focused on is preparing us for the event that we do get an incursion. So we always hope that it doesn't happen, but we do know that from time to time we will have those incursions. So we're focused on building the capability and capacity of Biosecurity Tasmania to support those responses, but we also work with industry, so TIA, Tas farmers, to sort of build capability and capacity across the industry as well. But in addition to that, there are things that can be done on farm. So it might seem like a small drop in the ocean, but it's really, really important. One of the key things that you can do is have an on-farm biosecurity plan. So that's really important that you've got that. If you do have one already, make sure it's up to date, and then of course make sure you're actually implementing that plan. There is some funding that's been provided to Tas farmers to help farmers to actually prepare those plans, and that's a really great resource to sort of help you to pull those together. Biosecurity is leading as well in awareness campaigns around that key messages, come clean, go clean, that really help us from preventing pests and diseases in the first place, as well as stopping the spread sort of between farms. You know, it's really important that those messages are heard, particularly by contractors and people that might be moving between farms on a regular basis. One of the other things that's really important that BT's doing is supporting those avenues for reporting. You know, Doug mentioned earlier, there was that one person who saw something unusual in a potato, reported that through to Biosecurity Tasmania, and so we were able to pick up on that detection of PMTV. So if you see something unusual, it might be nothing, but it might be something. So there's a number of sort of hotlines that BT manage that can be used to report those when you do see something unusual. There's a heap of other stuff that Biosecurity is doing in collaboration with industry in the sector, sort of contributing to preparing for incursions that might come our way. You know, these can have real big implications on the agricultural sector, and you know, we really want to maintain that reputation of, you know, a real foodie destination. And yeah, it's really important that we sort of all work together and play our part in that.
Yeah, thank you very much, Alice. Craig, I've got you now. What is a biosecurity engagement lead? What's the purpose of your role, and how do you see that fitting into what we do here at TIA?
Yes, I suppose, what do I do? A little bit of the glue between industry, government and the research is probably the main part of it. So we're sitting down at the moment working through what exactly are the problems that we need to face, both as researchers, but also with that extension and engagement role. So it's easy to get overwhelmed with the number of different pests and diseases out there. You've heard from Doug and Calum just about a small sample of them. Go across the different crops. I know TPP was mentioned, tomato, potato, psyllid. That's, again, unfortunately, a matter of time. It's not under control in Victoria. It's been detected in the outer suburbs of Victoria just a month ago. It's in the Bellarine Peninsula, so it's close to one of our entry points as well. Leaf miners are coming further south. Fall armyworm has made it to Tasmania once. At the moment, climate is working in our favour, but that's a changing situation that we'll be facing. So add to that something like Xylella. I think you might have got to mention in the news today about a change in a risk assessment. That one's going to be a landscape changer. So it's a plant bacteria, affects about 900 different plant species. It's all through Mediterranean Europe, North America, South America, and affects a wide range of different crops. So you start weighing all those up and you go, yeah, biosecurity, it's bad news. But what my role is looking at is, what is that system? How can we improve the system? Where's our gaps or our vulnerabilities in here? And work with groups like BT to say, how do we look at those pathways for entry? How do we improve surveillance on it? How do we prevent it both getting into the state, but also onto properties? So as Alice said, the importance of our farm biosecurity plans, and then actually use them. It's one thing to have a document saying, hey, I'm going to check everything that's coming onto my property, but how easy is that to do? How do you actually do it to actually try and minimise your risk on there? And that's the way we're approaching it, looking at both that prevention side, work with BT around the preparedness side. Part of TIA's role is providing support during emergency responses, so we're looking at training of our staff as well to provide that support. But then during the response and during that recovery phase as well, of how do we get people back on their feet, what sort of support can we actually do? So we're looking at other providers as well, how we provide them with support, awareness, training, so they understand what their role is inside that biosecurity system. And so we're looking at that, both at the pest, host, vector sort of relationships, as well as the pathways, but also what that actually means in the changing and natural environment. And importantly, we understand biosecurity's a big part of it is about people. So looking at that social element of how do we get people motivated and understand what they need to be doing, what they need to report, and how do they need to react when they do detect something that's a little bit unusual, and then make sure that any of those barriers that make it harder for people to do biosecurity, we address those or try and remove those. So there's plenty to happen. We're in the final stages of developing our strategy and what some of our priorities are, and we're talking with BT and some of the peak industry bodies about that now.
Yeah, that's right. And if you do get a chance to have a look at the QR code and see our strategy, you'll see biosecurity has a big part to play in it. And I think your role, Craig, is going to really fuel some of those key deliverables of that strategy. So, Doug, with your Forthside Farm Manager hat on, aside from avoiding sunburn, what would you say would be Forthside's major role here as a research farm to address Tasmania's biosecurity challenges?
Practices. Yeah, so, I mean, we're pretty lucky. So, I mean, lucky or unlucky, we're not a large enough scale to afford the sort of machinery that is effective and efficient. So we have a large number of contractors that come on site. With those contractors, they're absolutely awesome. They're very mindful around coming in clean and then leaving clean. Field officers, yeah, sure, they sign in, but again, they're very mindful around boots, use the washdown bay and everything like that. I suppose the question I'd throw back to the growers in the room to start off with is, since August, has your biosecurity changed on farm? I'm not going to embarrass you and say put your hand up or anything like that, but has it changed? I'd probably suggest for the majority of people, unfortunately, it hasn't. And I feel really bad for saying that, but I don't see it. I do occasionally, but not as a broader scale. And that to me leads to a lot of frustration. We've got Simplot and McCain's invested a lot of money in disposal of seed. We've seen Biosecurity Tas, state government, spend a whole lot of money on testing to determine where we're at. And yes, sure, we enjoy a low level of detection of mop-top, in this particular case mop-top, within the state. So what we do need to see is an improved uptake of biosecurity on farm, and I'm so genuine in that.
Yeah, thanks a lot, Doug. Calum, thanks for your presentation before. Keen to get your perspective on what you would say to growers who have concerns about mop-top on their farm and other diseases and how they can best be managed.
Sure. I mean, from a virus management perspective, I think Doug touched on it before. I mean, in reality, globally, the biggest viruses are PVY and leaf roll virus. They're both spread by aphids rather than a powdery scab management. And when you're talking about management, a lot of Tasmanian growers would turn around and say, oh, they don't exist here because they're so rare. The reason they're rare is because of seed certification. Management of viruses in general is manage the source, not the vector in general. And seed certification is a big piece. So in Tasmania, we have an amazing seed certification system. We're also isolated. And a lot of these sources of the virus disappear, so we actually don't bother about, and again, talking with John's presentation earlier, we don't use huge amounts of insecticides because we don't have to. Almost everywhere else around the world, they drown their potatoes in insecticides to try and stop viruses being spread by aphids. We let the predators do that for us and manage that. So not leaping into the chemicals shed to try and control aphids is for that. Mop-top, well, that's slightly different because we're talking about a different vector, and it's one that's we don't have terribly good controls for anyway. But once again, the source, the potato source is important. With mop-top, obviously, then powdery scab management, and part of that is understanding where it is on your property because it sits in the soil for long periods of time. So testing and there currently are tests for powdery scab that are effective, and you can map your farm and work out which fields are more likely to have more powdery scab or not. And in the short term, they'll be matched up with mop-top virus testing as well. So you'll be able to say, have I got the vector and have I got the virus, and then can make some decisions around that. Be aware and not frightened, I think, is the main thing. I suspect mop-top virus, it's a new incursion, it's a bit scary. It may not be as big an impact as it could be. And something like TPP, if that arrives, even without CLSO, psyllid yellows is a nasty thing we don't want. So there'll be unfortunately other things that come along which will cause us more issues. With TPP again, avoiding leaping into the chemical shed and trying to drown it in insecticide may well would be a nice approach, and actually see if we can use our existing IPM strategies to help manage these insect pests. Anyway.
Yeah, it's really great perspectives, Calum. I want to throw to the audience now. Does anyone have any questions for the panellists here? Please put your hand up and Lauren will pass you the mic and go for it.
Just a quick one to the panel. Given the pressure on our local seed producers producing for export and our fresh growers for potatoes, how long before do you think it will be before mop-tops positively identified on the mainland and some of those restrictions can be lifted and our farmers can get on with business?
I'll have a go for that one if you like. Depends when people start testing, I guess. Look. It's unknown. I mean, until tests are there. I mean, particularly soil tests for presence of the pathogen. I mean, that they are being developed, and once that's in place and industry start to test around the country, then we'll determine distributions. Is it highly localised? Is it everywhere? Good question. We'll find out, I suspect, over the next couple of years.
Question at the back there.
This one's either to Doug or Alison. Just wondering when the incursion first occurred, and I think there's some RNA testing that was going to be done on point of origin of this disease, because there were several, different pathogens around the world, and I was just wondering how that work was coming along.
So there has been some sequencing done. I am not 100% sure on where that is at the moment, but there is definitely work being done to determine where exactly this strain has come from. I don't know, Calum, if you've got any more to sort of add on that from your perspective.
I must admit I was meaning to try and get some more details before it came up. There has been some strain typing, and I can't recall exactly which strain, which region it was most closely associated with. What I do know is it's not the New Zealand strain, so it's quite different. And there's different mutations in the sequence, and it's clearly not what's present in New Zealand, which would have been an obvious candidate for leaping over the Tasman. But then again, movement of farm machinery or otherwise from Europe or North America. I mean, there's opportunities for anything where there's potential for even a small amount of soil to move. That's another way of things coming in. So I suspect it's more likely European, but I can't recall.
Any other questions from the audience?
Hi, it's Jodie from Agribusiness in UTAS. I've got a question about tomato potato psyllid. It being an insect, do you have any updates for us on what the biosecurity measures are currently being implemented to reduce the risk into Tasmania? I'm just thinking of the import requirements for a lot of our fruit and veg that moves into Tasmania.
I can provide a bit of advice on that one. So Biosecurity Tasmania has certainly increased our inspection of host material coming into the state. And there are also some import requirements, and I believe they are now in place or about to be in place, that put additional restrictions on material, both host and carrier material that's being imported from Victoria.
Any more questions from the audience? All right, I actually have another question for Alice. Alice, you talked about building capacity and capability in BT and also TIA. What does that look like? What sort of training is involved? How does TIA get involved in that? And what is that sort of relationship that BT and TIA actually have to deliver biosecurity response?
So as Craig mentioned, we are very lucky to be able to draw on TIA staff during the response. Earlier in the year, we ran a bird flu exercise, so sort of simulating what a response to a detection of bird flu in the state might look like. And we actually had a couple of TIA staff come along and participate in that exercise. So, you know, my expectation with Craig on board now is that we'll continue to sort of grow that relationship and build on that sort of training and developing that capability across our organisations, because, yeah, I think we'll be very reliant on TIA in the event of an incursion in the state.
Yeah, Craig, I know that you're going around to lots of the different peak bodies across Tassie and talking to them about some of their challenges. How do you see that relationship working with TIA in the future?
Essentially looking at us as a little bit of a problem solver, which, you know, we can't solve all problems for everyone, but just picking out what some of those key issues are. And just picking up what Alice was mentioning there, we've already had those discussions about what some of those training programs to look like, both, you know, formal training as well as working with them on exercises. So I think the other part of that is building on some of that liaison work with the industry groups. And I think Doug mentioned it earlier about himself and Bonnie being an industry liaison officer during the response. This is absolutely critical. And this is a key part just from, you know, over the 20 plus years I've worked in biosecurity responses, that we have industry inside the tent, literally there, providing advice to government. As I think Alice might have said something about, you know, not being an expert on potato crops. So often that's what will happen. There'll be government officers who are specialists in biosecurity generally on particular fields, and it's always the virus or the pathogen or the insect pest that we least expect that will be arriving. And that's where we rely on that industry expertise to come in and provide the advice, so we plan out how we can actually contain it, how can we control it, or hopefully eradicate it. So that's going to be a big part of what we do, as well as just building up that relationship. I always say it's really critical that we have the relationships before we actually need them. If we're doing this sort of stuff during an emergency, we are scrambling to stay on top of it. So we need to, you know, understand how each other work, where some of the vulnerabilities are, and then, you know, literally during that emergency situation, be able to walk in there and actually know who you're talking to and what their skills and expertise are.
Excellent. Thanks a lot, Craig. One more final chance for a question from the audience.
All of you mentioned biosecurity being important on the farm level, and Alice, you said about supporting farms individually to make their own plan, and Doug, you mentioned that that's lacking and that people are not adopting those plans. I was just wondering what that support looks like and why is it not being adopted widely?
We have industry groups, so RMCG, VegNet, those sort of people running programs in areas. So they've had one at Forthside, I think they've had one at Longford. Tas Farmers, I think, have conducted some as well. So I think the growers that are genuinely interested are going, participating, understanding the logistics and logic behind it. Unfortunately, it is time and dollars at the end of the day. So I'll pick on potatoes because it's nice and easy. When you've got a potato contractor going, trying to get in 10 hectares a day or whatever the magic number is, to go from one property to the next, to do a full wash down, decontamination, everything like that, you're talking multiple hours. So that's multiple hours that machine is not working. So unfortunately, yeah, in this particular case, that's the space we're in. The grower ultimately, the grower ultimately is not willing to part with the extra cash, or not in a position to be able to part with that cash to reward the contractor for the clean down.
I'll just jump in there and make the comparison to workplace health and safety. It's a similar sort of thing. We've now got a duty of care provision in the Biosecurity Act, just as we do for workplace health and safety. So this is what we want to create as normal behaviour, is people come onto a farm thinking that I'm a biosecurity risk. Same sort of thing. If someone walks onto one of your properties and sees a trip hazard or some sort of chemical hazard, do they ignore it? No, they take action. And it's taking time. I was involved in the original Biosecurity Act in Queensland, developing that, and that's what we actually borrowed. We picked up the Workplace Health and Safety Act and went, we want this in biosecurity. We rolled on another, more than a decade, 15 years since that first act came in. Tasmania's Act's very similar to the Queensland Act, which is similar to New South Wales, ACT. We're seeing this roll out across the country. We're still building up that culture and understanding that this is everyone's responsibility.
Yeah, thanks a lot, Craig. Look, I'd like to thank the panellists for their valuable insights into biosecurity and give them a round of applause.